Hello everyone, this is Rohan Meshram again, and in our previous lecture video we learned very basic concept in protein folding like Levinthal's paradox and Enfensen's experiment and what might be their implication in ab initio structure prediction. We concluded the previous lecture on the requirement of an efficient ab initio structure prediction algorithm. Now let us move on our discussion on more refined concepts that are required in understanding this ab initio ideology as such. Okay, so to begin, uh, you might want to first understand what are molecular simulation methods. Okay, so I have tried to discuss these things in our previous uh, protein modeling lectures when we tried discussing the implementation of RMSD in structural bioinformatics. Okay, so anyways, there is no harm in getting introduced with the new concept formally, right? So why do we need to learn this molecular simulation over here? Because this particular method is needed to be implemented for predicting the structure using ab initio ideology. So I think it would be better that we spend some time on understanding some basic concept of molecular simulation methods as such. Okay, so the basic idea in molecular simulation methods is, well, say for example, we construct a particle based description of our system that we are interested for investigation. Okay, and once we do that, we allow our system to propagate over some finite specified time. So the thing is that you cannot continue your simulation for indefinite time or infinite time. Okay, so you cannot do that. Your simulation should be for, say, some finite specified time. Okay, that is an important aspect of molecular simulation. Molecular simulation must stop at some time. Okay, so now once you propagate the system for some finite specified time, now what you would do is that you would observe how your system evolves over that time period, and your system is then recorded in something called as the trajectory. So however your system evolves during your simulation, you would keep track of the system in something called as the trajectory. So every simulation method must generate a trajectory that would give you an idea about what things happen in your simulation. Okay, so once you have a trajectory, what you usually do during these molecular simulation is that you take the snapshots of the system at some specified time intervals. We call such snapshots of your system as frames in molecular simulation terminology, or molecular simulation thesaurus or glossary. Okay, so every molecular simulation would end up generating a trajectory, and in trajectory there would be different frames that would correspond to the state of your system at some specified time intervals. Okay, so here is how things can happen. Well, say for example, if you plot the conformation of your protein and energy of your protein over, say, some specified time, you might end up with such a type of graph. Okay, and we'll say for example over here we have time. This is the energy and conformation of protein. Okay, so now what you can do is that whatever simulation that you have conducted, you can then select at specific interval of time ki, okay, this well, I would save, well, say for example at this particular point your protein might look something like this, and at this particular point after some time your protein might look like this, and at this particular point your protein might have this configuration. Over here it can have this conformation. Okay, so at this specified interval of time, what you can do is that you can save the snapshot of your system. Okay, and you might be interested in say some interesting property of your system. Well, say for example you might be interested in finding out the free energy of your protein ligand system over say some specified time duration. Okay, so for whatever snapshot that you have taken at some specified time interval, in that snapshot you can calculate the properties of interest. Well, say for example in our case if it is free energy, then we'll say for example at this particular time frame the free energy was x1, at this particular time frame the free energy was x2, at this particular time frame the free energy was x3, and so on. You can calculate the free energy of we'll say for example at configuration time xn. Okay, so what you are doing is that in this way we say that we are sampling the structures in your trajectory. Okay, and whatever relevant properties that that you are interested in, you would kind of average all those property x1 to xn. Okay, and you can in this way you can analyze your system based on say some properties of interest like binding, free energy, and so on and so forth. So all in all what I want to say that some relevant properties of the system can be calculated for each frame that you have saved, and you can average it over the entire trajectory. Okay, so things will become more clear when I show you some of the MD simulation trajectory. Okay, so the frames will say for example I would just show you. Okay, uh, this is what we do in molecular simulation. Okay, now I will this is just a sample output of a molecular simulation that one of my student have performed in her post graduation study. Well, this molecular simulation is molecular dynamics simulation of say some chalcones with an enzyme called as cyclooxygenase. Okay, so this is how you obtain the trajectory. What you are visualizing is the trajectory that shows you how the system has evolved over the time span of 50 nanosecond. Okay, and every time the structure you see it is moving, you are actually looking at the frames that are placed one after another. Okay, and this is how you might end up visualizing a trajectory after performing say some molecular simulation. Over here this is an output of a typical MD simulation. Okay, let us move on. That now based there are two main categories of molecular simulations. Okay, and you can categorize this molecular simulation based on how molecular system is propagated in time. Okay, so if you use if you propagate your system deterministically, the molecular simulation that you are doing, we say that you are performing molecular dynamics simulation. Okay, and if you use the probabilistic way to propagate your system, we say that you are performing Monte Carlo simulation. Okay, well, let's try to differentiate between these two categories of molecular simulation. Okay, so the very first difference between this MD simulation, and this Monte Carlo simulation would be uh, uh, the way in which these simulations are carried out. Okay, so one would use this MD simulation in order to investigate the structural, dynamic, and thermodynamic properties of the system of interest. Okay, you would use this MD simulation to observe how the system has evolved. You would check. You would be able to perform. You would able to analyze the dynamics of the system, as well as you would be able to perform, or you would be able to figure out the thermodynamic properties of the system. On the other hand, Monte Carlo simulation are used to investigate only the structural and thermodynamic properties of the system, but they cannot be used to infer the dynamic properties of the system. Why is that? It would clear out in a little bit while. Okay, so as I have already mentioned, deterministic rules are used to generate a new configuration from the present configuration. Okay, so every time, if this is the initial state of the system, and after some time you would calculate the state of the system after, say, when some delta t time has passed. Okay, so whatever new configuration that is generated after, say, some specified time interval, what we call it as time step. So uh, to do this, we use something called as the deterministic rules. Okay, and if you are using this deterministic rule in finding out what might be the new configuration look like, then you are performing MD simulation. Okay, while over here, probabilistic rules are used to generate a new configuration from the present configuration. Okay, so we'll say, for example, if we speak in practical terms in protein modeling, this MD simulation or deterministic rules based simulation method are ideal when you have a folded state of a protein. That is, you already know that this is how the protein would fold up, and if your objective is to just monitor how structural changes happen in that protein over the specified period of time, then this MD simulation would be the method you would be looking for. Okay, but on the other hand, in protein modeling, if you using this MC simulation would be more ideal when you have a condition like you start out with, say, some random state of a protein which you know is not a folded state, and your objective is to find out as to what new configuration this random state might acquire. Okay, so this is the basic difference between MD simulation and Monte Carlo simulation. Okay, your MD simulation would generate something called as a continuous dynamic trajectory of the system, while the trajectory that is obtained in Monte Carlo simulation cannot be considered as continuous trajectory of the system. Okay, why is that? Well, say, for example, when you generate the continuous dynamic trajectory, okay, when you generate the continuous dynamic trajectory using MD simulation, it is said that the trajectory that are generated in MD simulation includes the concept of time. Okay, as we know that the simulation starts at some point and it would end at some point. Okay, well, that is not the case in MC simulation. Okay, in Monte Carlo simulation, the trajectory do not include the concept of time. Okay, so whatever trajectory you might visualize in MC simulation or Monte Carlo simulation would represent an ensemble of configuration that could be structurally sampled. Okay, so whatever frames in MC trajectory that you would observe, that would correspond to the state of the protein that are thermodynamically and kinetically accessible. and this would not mean that these states will be achieved in a given order as they appear in the trajectory. Okay, so things would become more clear when I show you a typical Monte Carlo simulation trajectory. Okay, so now based on whatever discussion that we have done, now I would like to tell you why this Monte Carlo simulation is more appropriate methodology to be used in ab initio structure prediction. Okay, so the very first feature of this Monte Carlo simulation method is that it allows you to pick up some random moves and don't necessarily require to know where your simulation would start and where your simulation would end. And this is the exact feature that we are looking when we are trying to predict the structure of a protein in silico or using your ab initio method. We really don't know from where exactly your protein would start folding and where it would stop. Okay. Moreover, your Monte Carlo simulation allows you to try random things and you can see what works after doing or after taking those random moves. Okay, so this feature is also necessary so far your ab initio structure prediction method is concerned. Okay, why? Because in your ab initio algorithm we shall require to take a lot of random moves while folding your protein. Okay, and your MC or Monte Carlo ideology supports such feature. Okay, and since we are dealing with a lot of probabilistic ways of propagating your system, and we would be dealing with a lot of random moves, therefore such a type of methodology are also included in something called as the stochastic algorithm. Why? Because it is involved, because this algorithm involves, it is complete, it depends on a lot of probabilistic moves or random moves. Therefore, it is included in stochastic kind of algorithms. Okay, so uh, here is the typical trajectory that you might end up when you try to predict the structure of a protein using ab initio method. Okay, so here is the trajectory. Okay, now you can see that this is a typical trajectory that you might obtain from Monte Carlo simulation. Okay, so what you see over here, the frames that you see in this MC trajectory correspond to the state of the protein that are thermodynamically and kinetically accessible. Okay, so whatever state that you have seen frame by frame, well, that do not mean that these states would be obtained obtained in the same order that they appeared in the trajectory. Okay, so whatever samples that you have whatever structural states that you have sampled, that you see one after another, well, that do not necessarily mean ki they would happen in that particular order or this particular order. Okay, just some these are just some structural state that are accessible by this protein sequence in a given time frame. Okay, and that is why we say that these trajectories are not continuous, and your MC simulation do not have the concept of time. Okay, these are just ensemble. Ensemble means what? Collection of, say, some structural states. We call it as ensemble, and it is not necessarily that whatever structural state that you have generated in this trajectory would end up continuously in, say, some specified given of time. Okay, so there is no concept of time in Monte Carlo simulations. Okay, now I hope you understand this concept. Okay, so we would be revisiting this particular video multiple times as we move on to uh, understand the concept of this Monte Carlo folding algorithm in more details. Okay, so uh, whatever I am going to kind of discuss in this lecture is based on Kim Simons and David Baker's approach that were designed back in late 90s. So I would advise you that you read these 8 papers if you are interested in going in more details of this Monte Carlo prediction algorithm as such. Okay, so this is where you would want to go in more details of. Okay, so read these papers if possible. Okay, so now let us discuss what is exactly the Kim Simons and David Baker's approach for predicting the structure of the protein using ab initio methods. Okay, so there are two main concepts in the series of papers that I have just shown you, and basically Simon and Baker's approach is based on these two main concepts. Okay, so the very first concept that these guys relied on was that the local sequences gives a bias towards small number of local confirmations. Okay, well, what do we mean by that is that local sequences, well, so for example, if this is the protein that you are dealing with, then a small stretch of the peptide sequence or small stretch of your protein sequence, we would call it as local sequence. And we'll say, for example, if it is a 9 residue stretch, we would call it as 9-mer stretch. Okay, so now this local sequence, like this 9-mer stretches that we are talking about, or what we call it as the subsequence of the protein, well, structurally, these local stretches have more tendency to adopt either one of the secondary structure rather than trying out all possible conformation. That is the main concept. Okay? If you consider a 9-mer stretch, this 9-mer stretch is usually biased towards one of the secondary structural state, either helix or strand or coil, rather than trying out all of the 3 structural states. That is what this statement means. The local sequence gives bias towards the number of local conformation, towards a small number of local conformations. Okay, and the second important concept is that whenever the protein folds, okay, there are some non-local interaction that would preferentially stabilize the native global like structure. Non-local interaction will say, for example, this can be hydrogen bonds or van der Waal contacts and so on and so forth. That can be included in non-local interaction. Okay, so the concept is that the non-local interaction would stabilize a native like global structures. So these are the two main concepts based on which this Kim Simons and David Baker's algorithm work. Okay, so that is that is one important concept that you should know. One more thing that I would like to clarify is that we need to delineate between what is ensemble approach and what is optimization approach before we proceed for the Monte Carlo uh, structure prediction algorithm. Okay, so uh, well, what is an ensemble? As I have already mentioned, in Monte Carlo simulation or MD simulation, you would end up calculating the trajectories that would tell you how the system has evolved. Okay, so these trajectories are nothing but the collection of various state of the protein that would fold into. Okay, and what we can do is that we can assign the energy states as I have told you to all the states of the protein in an ensemble. Okay, so now the idea is to kind of to capture and understand what is the energetics that is going on in an ensemble. If you want to capture the energetics of an ensemble reasonably, you would proceed to something called as the ensemble approach. Okay, on the uh, well, say for example, you can kind of find out some average thermodynamic properties using this ensemble approach. Okay, so well, say for example, as I have already discussed, that if one wish to find out the free energy of a protein at a particular set of condition, okay, one would kind of sum up all the high energy and low energy configuration from your energy profile and get an idea of what might be an average energy of the whole system as such, or the whole ensemble as such. Okay, on contrary, when you talk about performing an optimization, in this optimization approach you would be interested in finding out that one lowest energy conformation from an ensemble. Okay, so in protein structure prediction scenario, we are not interested in all the different structural state that the protein can acquire in the trajectory. We are interested in, let's say for example, we do care about that one state in that particular in that ensemble, that one state that dominates the ensemble rather than the other states that it might predict. Okay, and this one state that we are interested in might be the folded protein conformation we seek to find out. So this is the basic difference between carrying out the ensemble analysis versus carrying out an optimization analysis. Okay, so in this way, in optimization, we focus when we take this optimization approach, what we say that we focus on very few states and not on the 10 raised to power 143 state that the protein can acquire. Okay, our objective is, our idea is that we are interested in finding out the lowest energy state. Okay, and we shall go on discarding the conformational state of a protein if they are higher than certain threshold as compared to previously found low energy state. Okay, so so now it is time for us to again get back to our simulation, Monte Carlo simulation trajectory, and try to correlate what we have just learned. Okay, so so whatever frames that you would see in this trajectory, uh, each and every frame would correspond to the lowest energy state that is observed so far. Okay, so over here at this point, this is the lowest energy state that has been observed so far. Now, the moment you proceed further in your Monte Carlo simulation, and the moment new lower energy state is found, the previously identified step is discarded. Okay, so at this stage, this is the lowest free energy conformer that you have identified so far in the trajectory. Okay, now the moment you obtain a conformational state that has got lower energy than this one, you would discard this state and move on with the new state as such. Okay, and hence you go on finding out the newer conformations of the given protein. Okay, so that is what we do in this Monte Carlo simulation approach by implementing this optimization way of finding out the lowest energy conformer. Okay, and that is what we do in Monte Carlo folding algorithm. Okay, so enough of background is this is what you need to know before we proceed for understanding this Monte Carlo folding algorithm. So now you can refer Kim Simon's paper in 1997 for whatever is going to be taught you in next few slides. Okay, so now how this Monte Carlo folding algorithm works. Okay, so this Monte Carlo folding algorithm is based on fragment insertion method. Okay, so what is this fragment insertion method? So in this fragment insertion method, what you do is that you begin with the initial protein sequence, which you would consider it as unfolded state or a random state. We can call it as a linear state. Okay, and in this unfolded state, the dihedral angles of your protein backbone, that is phi, psi, and omega dihedral angle, they are about 180 degrees. Okay, so what we do next is that we select some random stretch at, say, some random position all around the stretch of your protein sequence. Okay, well, say for example, you have selected this random stretch now. Okay, so what you do next is that what you do next is that now just try to remember that what I have said in the postulates, or you can say the basic concept of this Monte Carlo simulation algorithm by Kim Simon and David Baker. What was said that the small local stretch or local region has bias towards specific secondary structure. Okay, so now based on this concept, what we would do is that we would decide what this particular stretch is particularly biased upon. Okay, what structural state do this small stretch is biased on? Okay, now this bias can be calculated by selecting a local structure from pre-calculated library of local conformation. So what you would do is that there is a pre-calculated library of local conformers, as you can see, for small stretches. Okay, so now what you do next is that what you would select one of the local conformer from this pre-calculated library, and you would calculate or you would copy the coordinates of this small stretch, and you would assign those coordinates to your protein sequence stretch as such that you have selected in your random move. Okay, you might end up something like this. Okay, so well, so for example, you have figured out that your this small stretch is biased toward this secondary structural state. What you have done is that you have calculated the coordinate of this stretch and assign those coordinate to this much region in your protein sequence. Okay, and hence you get hence you see that your protein has folded at this particular small stage. Okay, and you would again repeat the same procedure, again calculate, say, some random stage in your given sequence, find out, decide where this bias is. Okay, and find out what might be the local conformer that it is biased towards it, copy the coordinate of this local conformer, and again assign it to your stage of interest. Okay, and try to have, say, some different fold this time. Okay, and this procedure is repeated one after another for many iteration. Okay, that is in concept what we do in Monte Carlo folding algorithm. Okay, so in general, your Monte Carlo algorithm starts with making a random move. Okay, so well, say for example, we take those random moves to generate something called as the test conformer. Okay, what we do is that we pick up at some random position in your sequence. We pick up at some random position in your sequence, take a random fragment from a pre-calculated library. Okay, and what we do is that after we take that random move, we would end up in, say, some conformer. Okay, so in your next step, what you do is that you evaluate the change in free energy after making that random move. Okay, and how do we calculate this change in free energy? We calculate by subtracting the energy of test configuration from your starting configuration. Okay, that is what we do. So if this is the initial configuration, okay, and after taking the random move, you end up with something like this. Okay, so what we do is that we try to subtract the energy that is present in test configuration from your starting configuration, and in this way we calculate something called as delta E. Okay, so what we do in such a way is that we evaluate the change in free energy after making this particular random move by subtracting the energy of the test configuration from the starting configuration. Okay, so over here by calculating this delta E, we answer the question if the energy goes up or if the energy goes down when we replace this small fragment with the stage that we have begun with. Okay, so that is what we do. Okay, so now there might be two conditions at this state. Okay, after calculating delta E, if you find out that the change in free energy is less than 0, that is if the energy goes down, we accept this new test configuration. Okay, and we move on taking say some another random point or random stretch in your next MC round. Okay, now if the energy is equal to or greater than 0, that is if the energy goes up, we do not necessarily discard the configuration. Okay, what we do now is that we generate a random number R. Okay, we generate the random number R, and the value of this random number is kept between 0 to 1. Okay, and after doing this we perform some calculation on our observed test configuration, and after those calculation, if the value is found near to 1, we would accept this test configuration even if the energy goes up. And if this the test configuration after calculation, if it nears to 0, then we eventually reject that configuration. Okay, so now let us try to discuss what calculations are needed to be done in order to decide if we accept the configuration or reject the configuration if the energy goes up. Okay, so this is what we are talking. Okay, if the energy goes down, we would accept the test configuration and we would begin our next Monte Carlo round using the test configuration. Okay, but if the energy goes up, we do not necessarily round up discarding that state. We compare it with we generate a random number that is that lies between 0 and 1. We perform some calculation. Okay, and after performing that calculation, we would decide if we accept the configuration or we reject the configuration. Okay, so what exact calculation do we do? That is what is meant to be understood from this slide. Okay, so what we do is that we compare the value of R with the exponent of delta E divided by KT. Okay, so this is how you read it. If the random number is less than the exponent of minus delta E by KT, where this T stands for the temperature of the system used in MC simulation and K stands for the Boltzmann constant. Okay, so this is what we do. We say that if the random number is less than, if this random number is less than the exponent of this delta E divided by KT term, then we keep the configuration, we accept the configuration, otherwise we would reject the configuration. Okay, so now this equation or this expression don't come from nowhere. Okay, so it comes from statistical mechanics. It has to do something with the probability of seeing one state of a protein relative to another in a thermodynamic temperature equilibrated ensemble. Okay, let's not go into more details of the statistics here. It would be enough for you guys if you understand that this expression has a very firm background of statistical mechanics. Okay, so now the reason behind as to why do we do this comparison with R will clear soon in upcoming few slides. Okay, so this is what your algorithm is. We take a random move, okay, start with say some state, okay, then after taking the random move, we would evaluate the change in free energy, that is we calculate the delta E, okay, by subtracting the energy states from test configuration and the initial configuration. Okay, and then we make a decision. If the energy goes up, we would if the energy goes down, we would accept. If the energy goes up, then we would kind of compare it with R and eventually decide if we accept the configuration or we reject the configuration. Okay, so while doing this, simultaneously you do bookkeeping. This bookkeeping is very important. Why? Because we need to keep tab of how many configurations we have observed so far that has got the lowest minimal energy associated with it. Okay, so what we are doing is that we are looking for a minimum energy configuration for our protein, since we are using this optimization methodology. Okay, and if the energy of the test configuration that you have ended up is less than the energy of the lowest minimal energy configuration that have been observed so far, what we would describe in terms of E mean, then we store the confirmation as a minimum energy confirmation that you have seen in trajectory. Okay, and again we would move on for making the random mean, for making the random move. So that is what we actually do. We make a random move, generate the test confirmation, evaluate that test confirmation in the form of energy term, decide whether to keep it or whether to reject it, then check if the new configuration is having lower than the lowest energy state that we have encountered so far. Okay, and again repeat the step again. Okay, and in this way you can move back and forth and see as we saw in that movie I have shown you. Okay, so well, now it's again, let us revisit the movie that we have seen and try to correlate again what we have learned in details of this Monte Carlo simulation method. Okay, so now here is what is happening. Okay, so now when you begin your simulation, what you do is that you take a random move. We'll say, for example, over here you have taken a random move and you have generated this new configuration. Okay, then you test it on the energy term and decide whether or not you keep this new configuration. Okay, then you go to perform that bookkeeping, wherein you try to figure out if whatever new configuration that you have observed, do it have the lowest free energy that has been encountered so far. If that is the case, then you would keep this particular configuration and move on taking another random move, and hence you repeat this again and again, go back and forth this way, and this is what you see now in this trajectory. Okay, so this is how you kind of generate a lot of decoys in your Monte Carlo simulation method. Okay, so this is what is happening conceptually. Okay, what is happening is that, again, let us draw our energy landscape. On x-axis here would represent different conformation space of the protein in time. Okay, so when we make a random move, the energy changes that correspond to the peaks. Okay, the energy changes that correspond to the peaks is in this energy landscape. Okay, so suppose that this is the state, that this is the point where we might have started. Okay, and then what you do is that when we make a random move and calculate that delta E, and we'll say, for example, this is where you end up in your energy landscape. Okay, so next what you do is that you calculate this delta E. Okay, in this move the delta E would be negative, the energy went down, therefore we keep this particular state of configuration. Okay, then we move on to take the next move. Again you can see the energy goes down, we would again keep this new state. Okay, and when you take the next move, when you take the next random move, the energy might go up. Okay, so now we won't reject the new configuration, but we would calculate and compare the value with R. Okay, so therefore using this comparison of the energy state with a random number R to the exponent of delta E term, we can kind of climb up the energy landscape with small steps if you get a good set of random numbers. Okay, so this is the implication of comparing the comparing the energy delta E term with the you know uh, uh, with the exponent of delta E divided by kt. Okay, so this approach would also enable easy sampling of low energy state step by step. Okay, like this. Okay, so that if you follow this step and if you follow the Monte Carlo protocol step by step, and you end up kind of sampling the states that are in order of decreasing free energy, you are completely capable of reaching some global minima in the energy landscape. Okay, so this is one of the basic concept behind this. Okay, so this is how your Monte Carlo simulation or Monte Carlo folding algorithm works. Okay, I hope you understand. In next lecture, we would try to understand what are the tricks for minimizing the Monte Carlo simulation approach. Okay, so in this way, now you can imagine that this can be computationally intensive. Why? Because every time you take a random move, the energy calculations are needed to be performed. Okay, and this can be time consuming as well as this can be memory intensive as well, so far the computational resources are concerned. Well, in order to tackle this situation, people have figured out some way around. That is what we are going to discuss in our next lecture. Okay, so enough for this, enough for today. Okay, see you guys in your next video lecture. Okay, thank you.